Our first presenter this evening is Dr. Matthew Chahati. Dr. Chahati has a PhD.
Cserhati.
Chahati.
Cserhati.
Chahati.
Cserhati. It's a Cserhati.
Okay. This is him. I apologize. That's terrible. Okay. I do as a PhD in bioinformatics and a Bachelor of Science in Software Development from the University of Szeged in Hungary. Also an MA in Religion from Greenville Presbyterian Theological Seminary. It's been an active creation science researcher since 2007, written 45 plus creation science papers, speaker for Creation Ministries International from 2019 to 2020, worked at several academic positions, and one of his major accomplishments is the assembly of the Neanderthal and the
Denisovan genome.
Denisovan whole genome sequence, active in molecular barominology research. So I'm going to stop mumbling and turn it over to the man who knows.
Okay. So greetings, everybody. Can you all hear me? Okay. Okay. So greetings, everybody, and it's nice to be here in Wisconsin. I remember that my first time at the Society of Creation was way back in maybe 2015-ish. That was as a participant, and it's really great to be here now as a speaker. And so, just as Gary said in his presentation, that in my talk, I'm going to present positive evidence for creation. We're going to talk about the science of baraminology. It's an independent science in creation science. We don't rely on evolution. It's a self-contained science. We have our own equations, statistical models. It's an independent science in its own right.
Say there doesn't seem to be enough volume.
Yeah. I don't know if I could increase the volume here.
Sorry, let me break down.
Can you hear me? Okay. Yeah. Maybe I should put the microphone higher up on my lapel. Okay. Maybe I should use a handheld like this. Okay. I'll do that instead. Right. Okay. So now I'm here as a speaker. It's great to be here, and greetings to everybody in the audience and also online as well. So as a bit of a personal introduction, Gary told what kind of degrees I have, also what kind of papers I have. I also have several grants from the Creation Research Society, and some of the papers that I wrote in these creation science journals, 5 of them are stealth papers, meaning that previously I published these papers in secular journals. BMC Genomics, I published 2 of them. So just to show you that as creationists, we do real science, so much so that even though I have to use different words when I write the paper, even secular evolutionary scientists have to accept the science that I write in my papers. I'm also a member of the Creation Research Society, the International Society for Computational Biology, and the Evangelical Theological Society. So the outline of my talk is just an introduction to baraminology. What is a science? What is the basis for the science? Some of the terminology, some of the words and the scientific phrases that we use when we talk about baraminology, and also how we do baraminology studies, some of the methodology involved. So the whole science goes back to Genesis chapter 1, verses 20 to 21. And here we read, "Then God said, let the waters abound with an abundance of living creatures, and let birds fly above the earth across the face of the firmament of the heavens. So God created great sea creatures and every living thing that moves, with which the waters abounded according to their kind, and every winged bird according to its kind. And God saw that it was good." So notice here that the Bible doesn't speak about individual fixed species, doesn't mention species with two Latin names. We shouldn't force our modern view of taxonomy onto the Bible. Rather, we should let the Bible speak for itself. So here we mention animals, but Genesis also mentions different kinds of plants and the vegetable kingdom. In verse 12, we read, "And the earth brought forth grass, the herb that yields seed according to its kind." There's that phrase again, according to its kind. "And the tree that yields fruit, whose seed is in itself according to its kind. And God saw that it was good." So it's very important that we see that the Bible talks about different kinds of organisms, not about fixed species. It's a very big misunderstanding that very many times secular scientists have about creation science. So where does the word Barominology come from? In creation science, we want to honor the naming of our science. We want to honor God with how we name our science. And so instead of using secular names, we use two Hebrew words. Bara, which means he created, as in Genesis 1, chapter 1, Bereshit bara Elohim v'hashamayim v'ha'aretz. Bara means he created. Min means kind or type or like a group. Logos in Greek is science or knowledge. And so therefore, Barominology is a science of the created kind. So that's where we get the name of the science. In other words, we can call it biblical taxonomy, discontinuity systematics, where we look for differences between kinds. We don't look at continuity. We don't seek how to connect species together. Rather, we see real gaps between different groups of organisms. And so basically, a Baramin is a created kind, as we saw in the two passages from Genesis 1. So type, group, class, or cluster. Now, in taxonomy, what is a kind? What's the taxonomical level of a kind? It's about the level of a family. It can be a little bit maybe beneath or maybe above the family. So here, if you remember your high school biology class, we have, if we classify humans, human is Homo sapiens. The genus is Homo. We are from the family Hominidae. We are from the order of primates, together with monkeys, chimpanzees, old and new world monkeys. We belong to the class of mammals. We come from the phylum Chordata. We are chordates. We have a spinal column, and we also belong to the kingdom of mammals. But as the blue arrow designates, this is roughly where the level of the kind is. In my studies, I've seen that, for example, it may be maybe at the level of genus, maybe even order. If we look at crocodiles, alligators, garbals, these all belong to the same order, but obviously crocodiles and alligators, they're very similar to one another. We suspect that crocodiles and alligators both belong to the same kind. So you can go even up to the level of order. It's around this level here, which is the boundary of the kind. So historically, Linnaeus, he never held to the fixity of species, but rather he saw relatedness of species within groups, a reproductive community. This is very important to note that since species they're not fixed, rather different species can hybridize with one another. And so Linnaeus here, he rejected fixity of species. Species can interbreed, their boundaries are fixed. Species aren't fixed, their boundaries are fixed, yet species can interbreed between these bounds. Humans don't breed with chimpanzees, but we have interbred with Neanderthals in the past because we share DNA with Neanderthals, and that can only be if we belong to the same kind. So Neanderthals are human. I can give a whole separate presentation about Neanderthals, since I helped assemble the Neanderthal genome sequence. So anyway, Barominology allows for natural variation within the boundaries of a kind. A Brahmin is a reproductive community. And of course, first they thought it was the genus, then they moved up to the family. They've even classified species from one genus, and they put it into another genus. This has happened very many times in modern taxonomy. So even so, the level of the kind is roughly the family. In modern Barominology, it can be maybe in the superfamily or the order, as I detailed previously. Modern taxonomical units are really artificial constructs. The name of the game in Barominology really is to, if you have a group of organisms, you try to classify each organism into its respective kind. And really, this is a basic mindset in a baraminology study. And so, just to show you that these animals, these hybrid animals, are real things. This is a tigon. It's a cross between a tiger and a lion. This is a walpin. This is a whale crossed with a dolphin. This is a beefalo, which is a buffalo and a cow hybrid. This is another hybrid animal. I think it's a yak and some sort of other animal. This is a grizzly bear, polar bear with a grizzly bear. This is a camel and a llama. Then we have a zorus, which is a zebra crossed with a horse. And then we have a coy wolf, which is a coyote crossed with a wolf. And here we have a bird hybrid. I just don't know what kind of hybrid this is, but it's like a goose hybridized with some other bird species. So just to show you that there is positive evidence for creation, and that the creation model really is a sensible model. It makes sense, better sense than the evolutionary model. And actually, just recently they isolated the DNA sequence from some archaic bear species, showing that the brown bear has hybridized with the polar bear. And of course, these little nuggets of information, these interesting findings that even secularists make, they come every so often, even in a secular literature. So science is interesting, and science supports the Bible, despite the evolutionary spin in the media. So anyway, so as I said, that the top model is incorrect. This is a fixity of species. The evolutionary tree is also incorrect. So rather, we have an intermediate model. We have the creationist orchard. And so here's the time of creation, the 0 time point. And these trees, these little branches, they are individual kinds. One is a cat kind, a dog kind, horse kind, whale kind, etc. And so we see that new species can arise in time. I mean, we simply observe new species coming about as just simply observed scientific fact. But of course, the creation model can accommodate such observations because we're scientific. So now let's move to the second part of the talk, where we talk about the terminology of baraminology. And so a holobramin, there's holos, which is a Greek word, and baramin. A holobramin is the complete exhaustive list of species that makes up a single baramin. So it's like a full roster of species of a given kind. In other words, it is a group that shares internal continuity, that is, each member is continuous with at least one other member, and is bounded by discontinuity with all other species on the outside. No member is continuous with any species outside that particular group. These are two very important things to understand in how we determine the holobramin. So the previous 2 points, and make a note of this, is that this is the fundamental tenet of baraminology, that if we have a holobramin, all the species are continuous with one another, and all species outside are discontinuous with the holobramin. So for example, there's the cat kind. All cats can hypothetically interbreed with one another, but they do not breed with dogs, nor with horses, nor with bacteria, nor with humans, nor with any other type of animal. Now the second main term, and there are 3 main terms. The second main term is the monobramin, is a group of known species that share continuity with regard to, without regard to discontinuity with other organisms. A monobramin is usually a subset of a holobramin. It is typically a lineage of species. It's like a group of species within the holobramin. For example, the larger cats like tigers and lions and leopards, or Neanderthals. The Neanderthals, they are a subset of humans. They have special morphological characteristics. They went extinct, but nevertheless, based on their whole genome sequence, Neanderthals are humans. They were just very isolated, and so they have some different characteristics compared to modern humans. So this is a picture of the cat holobramin. And so here we have the felis monobramin, Felidae, which is, the smaller cats. And so here we have different monobramins, smaller groupings within the cat kind. And these blue lines, they represent different hybridizations between individual cat species. This group here, there are no connections, but based on overall morphological similarity, we suspect that these other species they're related to the 30 or so cat species. And so again, here is your tigon. Here is a I think a cross between a tiger and a leopard. So just again to show you that there are we're talking about that there are real hybridizations going on between different cat species within the cat kind. The third main term we have to understand is the apobramin. An apobramin is a group of known species that are bounded by clear discontinuity without regard to inner continuity. In other words, they may be one or more complete holobramins, one or more kinds. But why is this important? These are animals that are obviously different from all other animals. The importance of understanding the apobramin is that if we're looking at a large group of organisms, we really want to separate some group of species which we definitely know are different from all other organisms, which we know can hypothetically contain one or more holobramins. So for example, we have bats, which are flying mammals. We know that bats are not related to any other kind of animal. They're the only mammals that fly. So the only question is, how many bat kinds are there? We know that bats, they're separate from all the animals. The big question is, how many groups are there within bats? Pinnipeds, these are marine mammals. Cephalopods, octopuses and squids. Birds are flying vertebrates. Turtles are also morphologically very separate and very disjunct from all other animal species. So we know that turtles are not related to any other kind of animal, but the big question is, how many turtle kinds are there? And that's one of the big research areas within baraminology. Now, some other interesting terms that we might want to learn about, which really aren't so important. The term baramin by itself, in general, can denote an apobramin, holobramin, or a monobramin. The archibramin is the very first individuals of the holobramin, created during creation week. For example, Adam and Eve are the archibramin. The paleobramin are extinct members of the holobramin. The neobramin are the extant members of the holobramin. The kynobramin are possible future members of the holobramin, which may arise in the future through breeding, mutation, or natural selection. So, for example, if we have some plant breeders and they want to make a new fruit species, they just breed two plant species together, and they have a new fruit species which they might want to breed for maybe its taste or for its special taste or whatever. There's the polybramin, which is a group of organisms coming from multiple bramins. So here we have our diagram. This whole thing here is the holobramin. Since the holobramin, it's the exhaustive list of all species in the bramin. Here at the bottom, we have the archibramin, Adam and Eve. In the corner, we have a smaller subset, which is a monobramin, for example, Neanderthals. Paleobramin are the extinct species or individuals such as Neanderthals. Neanderthals can be paleobramin or monobramin. The neobramin are the extant species of the holobramin, and the kynobramin are the future species of the holobramin. So just how we can visualize these individual terms that we just learned about. Also in baraminology, it's important to discuss the arc kinds, because in creation, God created different kinds of animals and plants, but we know that during the flood, the earth was repopulated from the ark, because different animal pairs went onto the ark, and they repopulated the whole entire earth coming out the ark. So we have to know about the arc kinds as well. On the ark, most kinds were represented by one male and one female. 7 pairs of each clean animal were also present on the ark. Some were needed for post-flood sacrifice, according to Genesis 8, verse 20. And also as the next speaker, Nathaniel Jameson, wrote about in one of his books, he found that the diversity of these kinds today are larger than all other kinds, which are represented by only one male and one female on the ark. So the 7 pair kinds are today much more diverse than other kinds, which are represented by only one male and one female. 8 humans made up the human ark kind, Noah and family. And so since each kind represented many, many different species, this is how all the animals could fit on the ark. So if somebody tries to corner you with a question, well, the ark, it's so small, and how could it hold millions of species that we see today? The response is that we can sort of compact and we can compress many, many species into one single ark, in one single kind that was represented on the ark. So Noah didn't have to take so many animals onto the ark. According to research by Answers in Genesis, there were about, I think, maybe 8,000 kinds present on the ark. Semi-aquatic animals such as seals, otters, whales could have remained outside the ark in the water. Plants could have survived on mats of debris in the flood water, since the flood destroyed a lot of vegetation, and there were these debris mats floating around during the flood, and other organisms could have survived the flood on these mats of debris. So here's a picture which really sums this concept up really well. So here we have in creation, this is a creation time point, and we have several kinds. We have the dog kind, the monkey kind, the Triceratops kind, lizard kind, etc. So they were created at the 0 time point. Then they begin to diversify, and then comes the flood. And as you can see, that only one pair made it across the flood. And afterwards, they repopulated the entire Earth from this one single kind. If we go back here, then there's this one pair of the given kind which survived the flood and then repopulated the Earth after the flood water subsided. So each kind survived the flood, but not all kinds survived until today. For example, dinosaurs, Triceratops, do not exist today. So here's another model that in creation, the orange circle represents a single kind. They go in time up until the flood, and then a smaller subset survives the flood and then repopulates the whole world until the modern present time. According to certain estimates, there were 55 dinosaur kinds on board the Ark, each representing between 10 to 30 different species per kind. So, for example, these are representatives of the Ceratopsia kind. These are the well-known Triceratops. And then we have these other species with horns, this big shield-like structure on their head, beaks. And so you can recognize that these animals, they're all very similar. They're all members of the Ceratopsia kind, which is now extinct. So this would be an example of a holobaramin, which consists of extinct species. So now we go on to the methodology of Barominology. And so being good, faithful Christians, we begin each study with the word of God, not with the concepts of men, not with our own human logic. We begin with the word of God. We see what the Bible says about a given group of organisms. Let's see what it says about bats or seals or whatever we're studying. So we look for the biblical data for any given group. Then from the Bible, we can draw some general categories. Plants can only have been created on day 3. Animals were created on days 5 and 6. So therefore, we know that, for example, Euglena viridis, which is a one-celled microorganism, evolutionists, they say that it's the common point between animals and plants, that it's a single-celled organism. It started developing in 2 different directions. One lineage led to animals, one led to plants. We reject this because we know that plants and animals were created separately on individual days. Flying and swimming creatures were created on day 5. Land animals were created on day 6. So therefore, we know that birds are not dinosaurs. Don't let anybody fool you. Secular scientists may come with these pictures that Tyrannosaurus Rex had feathers, it changes all the time. They just want to, popularize this idea with your kids, just to, convert them into evolutionists. Don't get fooled. Secular science changes all the time because it's the ideas of man, it's unstable. God's word remains forever. So when we analyze animals, another question is, could they have survived outside the ark? I did a study on pinnipeds, seals, walruses. Were they on the ark? Were they off the ark? They're marine animals. They could have survived in the water. So that's a big question as to deciding whether a given animal group had to have stayed on the ark, or could it have survived outside the ark in the water. Of course, it's always helpful if the given organism group is mentioned in the Bible. Many times it's not. Also, another question is, what about fungi, bacteria, or archaea, or viruses? These are microorganisms. Early humans didn't have microscopes. They would not have been able to observe such organisms. And so that's why we have the organo-substrate theory, which says, which is tied to Joseph Francis, 2009. There's a paper on Answers in Genesis website that you can read about this theory. So since microorganisms are not mentioned specifically in the Bible, therefore we have a sort of a freedom as to how we can talk about them in the creation model. Yet these organisms, they comprise the vast majority of living organisms on Earth. There are about maybe 10 or 100 times more microorganisms than are macroorganisms. Early humans did not have microscopes, and according to this theory, these microorganisms could have been created on day 3 when the dry land appeared together with plants. That's when the dry land appeared and when we had soil, and so we had all these microorganisms in the soil. That's what this theory holds to. And these microorganisms, they take part in many biogeochemical cycles, the carbon cycle, the nitrogen, and also the sulfur cycle. And so since these microorganisms, they're not really, you can't really examine them by morphological methods. You can't measure the tibia of a bacteria. You can't measure the cranial volume of, let's say, of a virus. They have genomes, they have DNA sequences. So we can study the genetic characteristics of these microorganisms, which will be the subject of my talk tomorrow, which deals with molecular baraminology. So stay tuned. So Bible tools which we can use. There are many Bible translations which we can use for cross comparison. So, for example, the New English Bible calls the ostrich the desert owl. So there are variations in the translations of a given Hebrew or Greek word. Use Bible dictionaries. The Theological Dictionary of the Old Testament is good. New International Dictionary of Old Testament Theology and Exegesis. That's a mouthful. There are concordances, and there are also many online tools that you can use, like Strong's Concordance. There are lots of tools out there for your use. And so baraminology methods, we will discuss morphological ones here and molecular baraminology methods in my next talk. We'll discuss hybridization, since I showed you hybrids of different animal species, demonstrating hybridization between two species is a very useful tool in baraminology. We'll talk about the character matrix, character weighting, entropy filtering, and then additive and subtractive evidence. So this is going to be a bit more technical in level. So if we go back to the fundamental tenet of baraminology, it says that members of a holobramin shows both continuity between one another and discontinuity with all other species. So kind resembles kind, where self resembles self. There's a technical glitch. Gary understands. So the kind resembles kind more than non-kind in a statistically significant manner. Monobramins have inner continuity without regard to outer discontinuity. On the flip side, apobramins have outer discontinuity without regard to inner discontinuity. So in this table, we see that the holobramin in green has both inner continuity and outer discontinuity. It's a full, complete species list. Apobramins, we're not sure what kind of inner relations exist within the apobramin, but we're pretty sure that it is discontinuous with other species, for example, bats or birds are good examples of apobramins. But the main question is how many bat kinds are there, how many bird kinds are there? We have to split up the apobramin. And also monobramin, the yellow, we know that monobramins they're continuous with one another, but we don't know whether they're continuous with any other monobramins. So with monobramins, it's sort of like a Lego puzzle. We have to piece together the species until we reach the level of the holobramin. So these are two different approaches as to how we approximate the holobramin. So let me just take a sip of water here. So a key methodology in Barominology is hybridization. It's when we discover different hybrids between different species within the group of species that are under study. So again, the biblical kind is a reproductive community. They reproduce amongst one another, but they don't have any continuity with any other species outside their own kind. Cats don't breed with dogs. So if we can show that two species have viable offspring, if they hybridize, then they belong to the same kind. For example, a tigon or a liger, which is a cross between a tiger and a lion. Now note that even if two species cannot hybridize, this is not evidence that they're not members of the same hologram. They may still be members of the same team. So two species may still hybridize with the same third species. So species A hybridizes with species C and B with C, then by transitivity, A, B, and C are in the same Brahmin. A with C, B with C, and so therefore A and B are in the same group with one another. If we look at the tiger and the house cat, we see that they're very different in size. I mean, there's a sort of a physical block between a house cat and a tiger having offspring with one another. Yet we know that that over time, since tigers interbred with other cat species over time and following different cat lineages, we see that in decreasing size, that house cats are still connected with tigers. It's just that they're removed from one another over several generations of breeding. There are also so-called crypto species, hidden species. For example, there are corals where one species it produces sperm, and the other coral species has an egg cell. It's just that on the egg cell and on the sperm cell, there's these receptors, and since mutations have destroyed these receptors, the sperm cell and the egg cell cannot join one another. It means that these two coral species are incapable of forming hybrids because the reproductive cells are incompatible with one another due to destructive mutations. But originally, they could have still belonged to the same hologram. And also years ago, creationists they devised a hybrid database which contained all sorts of data on hybridization experiments showing different levels of evidence for hybridization between different pairs of species. This here is a creationist hybridogram, which was created by the Hybridogram package in R. So just to show you that there are program software which are based on creation science premises. And so here what we see is we have different seal species, Phoca, Hybrida, Monaca, Cystophora, and these yellow orange squares, they represent different levels of evidence. That white means no hybrid, light yellow means assumed breeding with third species, assumed breeding within genus, breeding within the same species, documented hybrid, And so we can see that if we match up two species, we can see that this Phoca larga forms a hybrid with Phoca vitulina. They've actually observed these two species hybridizing with one another. But for example, yellow shows a weaker level of evidence of hybridization between two species. And so here we can see that there are different groupings of species into what are apparently different monogramins. So this is creation of software which visualizes relationships within Brahmins. We can also determine Brahminic membership without hybridization. In the case, if there's no hybridization evidence, we can show that there's continuity, or that is high level of similarity with other members of the same kind if we compare their morphological traits, or if we look at molecular traits, if there's high level genetic similarity between two species or two groups. You might have heard that they say that chimpanzees and humans are 99% similar. Creation scientists have shown that number is false. It's very lopsided. The similarity is more closer to 90%. On the other hand, we've shown that Neanderthals and humans are the one which are 99% similar to one another. So since Neanderthals and humans, they have a high level of DNA sequence similarity, Neanderthals and humans, they really truly belong to the same kind. We're both human. And also, we're going to look at molecular traits in my next talk tomorrow, so stay tuned. So another very important thing that we use in Barominology studies is a so-called character matrix, which is a collection of n morphological traits from m species, producing an m times n matrix, where the individual values are discrete values. 0, 1 represents the presence or absence of some sort of structure. For example, Japanese have an extra bone, and it's called the os japonicus. So 0 would mean the absence of this bone in non-Japanese people. 1 designates the presence of this os japonicus in Japanese people. 1, 2, 3, or whatever, it's a range of values. Question mark denotes missing values. We're going to talk about different weighting schemes, entropy filtering. And so, for example, this is an example of a character matrix where we have a species on the right side and we have traits on the top. So the individual columns, they are individual traits for these species which are in the individual rows. And so if we want to visualize Brahmins, in a simple example, if we have, let's say, only 3 traits, we can place all the species of the given study within a 3-dimensional cube, like so. So if we have 9 species, and if we're looking at height, width, and weight, in a very, very simple example, we can see that we have these three Brahmins, which are very tightly clustered to one another. However, if we study n different traits, if we study like maybe 100 different traits, we have an n-dimensional hypercube, which is hard to visualize. But so what we do, there are several mathematical statistical methods which can reduce different dimensions. The principal component analysis, or k-means clustering, may visually separate species from one another into individual clusters or Brahmins. Weighting schemes. It's important to talk about what type of traits are important, which ones are more important, which are less important. What about craniodental features? These are the skull and also dental characteristics. When we're looking at human fossils, very many times only the skull is present. We only have the skull. We don't have the postcranium. We don't have the rest of the body. So very many times in these Barominology studies, we're comparing skulls with skulls, like Hamlet. He's looking at these 2 skulls, and so which one is human, which one is non-human? We have to use a holistic approach. We shouldn't cherry pick individual characters which would support our hypothesis. This actually happens. Cherry picking happens very many times in evolutionary studies, which shows bias, which shows a religious commitment. Also, if you want to do such a baraminology study, also consult with paleontologists and also other biologists who can give you their expertise as to which traits are more or less important. So, for example, the width of a molar of one of your teeth is intuitively it's less important than the cranial capacity and should be given a lesser weight in a baraminology study. And also, the arrogance of modernity is inadmissible in baraminology. What do I mean by this? If we want to describe the whole entire human Brahmin just by looking at modern humans, that's an incomplete picture. We have to look at archaic humans who lived in the past, Homo erectus, Neanderthals, Homo heidelbergensis. We have to take the whole entire view of all humans into the picture in order to determine the baraminology of humankind. So we cannot judge the entire human hologram based only on what modern humans are like. Also, the Neanderthals, Denisovans, we have archaic humans who lived in the past. They're also humans. We also have to take them into consideration. We have to look at a Brahmin in its entire historical context ever since Creation Week. The next topic is somewhat technical, so if you don't understand it, that's not a problem. This is entropy filtering. Is when we filter our character set, because some characters might be problematic. What if we have a fossil primate which has a more or less complete postcranial skeleton, and we compare it to only skulls from other species? So we have, let's say, a fossil find where the skeleton is almost complete, and we have 9 other finds where we only have the skull. And so it's a very lopsided comparison. How do we do a baraminology study with such lopsided data? Should we retain the character values in this individual? What should we do? How should we possibly filter out some of the species or some of the characters? If we retain the postcranial characteristics of the complete species, we might reach a conclusion which would really separate this one species from all the other ones. It might be the case that they all belong to the same group, yet this more complete individual might be classified very differently because it simply has more data. So we have to filter characters described in this paper here that I wrote. So filtering criteria, and let me just have another sip. So we would remove columns or characters which have low entropy. And entropy, what we mean by entropy is variability or surprisability. More exactly, Shannon entropy. So what this means is here, if we have this figure here, on the x-axis we have probability of a given trait, and on the y-axis we have entropy. So what this means is if we have a coin, which is very lopsided, when you flip it, it gives 90% heads, 10% of the time it gives tails. So this is a very biased, unfair coin. So the probability would be, here it would be about 10%, and so it would have a relatively low entropy value. If we have a fair coin, which is 50-50%, here's 50%, and so the entropy value would be much higher. A fair coin is much more variable. It gives you a much bigger surprise. If you have a lopsided coin, you flip it many, many times, you get heads most of the time. It's not much of a surprise. It's not very variable. So we want to get rid of these kinds of characteristics. We're looking for these kinds of characteristics to retain in our character matrix. So we're going to remove columns with low entropy, remove species with too few characters, or too many compared to all other species. So if we go back to our data matrix, we see that we have these columns. We have a lot of question marks, not too much useful data here. We have this species J, which also has too many question marks. We also have this column here, and as you can see that it has a lot of 1 values. Here's a 0. So this column here, this would be like the falsified coin, which gives heads 90% of the time. We want to filter out this column, these columns, and also this row here. We want to get rid of them. So after filtering, we get this matrix here. And so the reason this is useful is because if we have a data, we have a data set such as this one. These are dinosaur species by Reese and Curry from 2017. If we filter this data set, we get this data set. And it's much more cleaner. There's less entropy in this data set than this one here. So this is much more cleaner, and the conclusions that we can draw from this data set are much more statistically, much more reliable. Another concept which is quite interesting is the potentiality region. The definition is that portion of the n-dimensional hypercube, if we're studying an n number of characteristics, and we have this hypercube, this big hypothetical hypercube. That portion of the n-dimensional hypercube where members of a given Brahmin can actually exist. So for example, it is biologically impossible for humans to be 1,000 feet tall. There are vascular constraints. We wouldn't be able to pump blood 1,000 feet tall, to a person who is as high as a skyscraper. It's just physiologically impossible. So each hologram has its own potentiality region, which does not overlap with the potentiality region of another kind. There is no human Z, for example. They actually did experiments in 1924. Soviet communist a scientist tried to cross humans with chimpanzees, and he was unsuccessful. There are real boundaries between chimpanzees and humans. So this is a picture of the potentiality region. When we look at just two characters, we find that there's this hologram, this other hologram, and this third one, A, B, and C. We can see that at this point, there's no connection between these two kinds. They simply don't overlap. So we see these three dots here. These are known species, but then there's this region outside these known species where there are unknown species. We simply either don't know, we haven't discovered these species yet, they haven't been hybridized, they haven't been formed yet. These are unknown species which can still arise in this portion of the potentiality region, but not in this region. These are impossible species. Todd Wood, who wrote a very influential book on Baraminology, for example, he says, can you think of a bookshelf bear? A bookshelf bear, it's an impossible species. It just simply doesn't exist. And so this region outside the potentiality region, it designates a space where species simply cannot exist, like 1,000 feet tall humans simply cannot exist outside their own potentiality region. One major way of discovering the hologram, starting out from the monogram, is using additive evidence. So we go from the monogram to the hologram. We can use successful interspecific hybridization results. If two species hybridize, they are definitely in the same Brahmin. For example, modern humans and Neanderthals, since we share DNA. You can do the 23andMe test. You can get yourself sequenced, and they'll tell you what percent of your DNA comes from Neanderthals. So we're all part Neanderthals. We can be proud of that. So there are also morphological similarity. Two species in the same Brahmin will very many times look similar and have similar genes. There are also stratomorphic species. There are fossil series which are connected by clear signs of character transition. So again, here we're looking at the monogram in yellow, and we want to approximate the hologram in green. So we're extending outwards from the monogram to the hologram. So what we do is we find one group. This would be a monogram. Then we add species which are close to that group. We have this group here, and then we add all three together. And so we're going from different monograms, we're glomming them together, and we get the hologram, which is this big group here. This is the additive method. There's also the subtractive evidence method. When we approximate the hologram from the appogram, it's the other way around. First, we want to glom groups together. Now we want to break things apart. So for example, I've studied the bat apobramin, and using molecular methods, I wanted to find how many bat groups are there within bats. There are megabats and there are also microbats, two possible candidates. But so here we're going from the holobramin to the apobramin to the holobramin. In scripture, if the two groups are created on separate days, birds on day 5, reptiles on day 6, we know for sure that they're definitely two different holobramins, because God did not create them to evolve between 2 days. That's not what happens. There's also morphomolecular dissimilarity if two species from different Brahmins look very different, or if they have very few genes in common, which will be part of my next talk. And also if they have unique synapomorphies, universally common traits within a group. Syn means together, apo means point, morph means trait. So if there's a lack of transition between the synapomorphy with other species, for example, they've never ever discovered something which is between a bat and a non-bat. Even the oldest bat fossils are still fully formed bats. Or turtles, there is no fossil turtle where the shell is only half formed. So we know that turtles and bats and birds are examples of apobramins. We just need to find out how many groups there are within these apobramins. Or there might be a presence of specific genetic markers or lack of a fossil continuity between invertebrates and vertebrates. So we know that vertebrates are another apobramin because there's a clear cut between them and invertebrates. So what we do here is it's the opposite way around. We start from the big group, and then we find that we separate this one species, then we separate this group into 2 other groups, and then furthermore we have these 3 groups. This would be the apobramin, and these would be 3 holobramins. So we're splitting up, let's say, bats into, let's say, megabats, and then Vespertilionidae and some other bat group. According to my study, there are 4 bat kinds, megabats, and then 3 microbat holobramins. And so we're going from the apobramin and we're going down to the level of the hologramin. And so lastly, I'm almost in the end of my talk, but here's a question for the audience. Are these 3 animals the same kind? And if anybody has a guess, just please voice your opinion. Are these animals the same kind based on what they look like? You're saying no? You don't think so? I'm sorry? You think yes? Any other opinions? So there's a yes, there's a yes, there's a no. You say yes. Okay, so several no's, several yeses. Now this is a fish, shark, it's a reptile, it's an ichthyosaur, and this is a mammal, it's a dolphin. These are levels of the class. So the class is even above the order, which is the upper boundary of the kind. These are different classes of animals. And so superficially, they resemble one another. I mean, they've got fins, they've got, you know, like a rostrum, they've got a tail, they have fins, they swim in the water, but they are separate kinds. They're not the same kind. Superficially, they might be characterized in the same kind, even though they're separate classes. But the big question is, how would we classify these animals based on their DNA? DNA, I mean, it's the hereditary molecule. You have to isolate the DNA. You have to run molecular algorithms to analyze the DNA and find whether these animals belong to separate kinds or not. So this will be the topic of my talk tomorrow. So please stay tuned. And so as a bit of summary and conclusions, there's been a lot of pioneering work done in classical morphological Baraminology, and it will continue to be used. And of course, morphological and molecular Baraminology can mutually support one another. However, morphology has its limitations, fragmented fossils, for example, only craniodental remains, superficial physical resemblances, for example, convergence. This is an example of convergence when you have animals in the same environment and they have similar morphological traits to allow them to swim about in the water. This is convergence. And so this is a limitation or high degree of sexual dimorphism when one sex is highly reduced as in organoids, when let's say the male is very highly reduced in morphology, very dissimilar to the female individual. Microorganisms, you can't measure the cranial volume of a bacteria. You need to study its DNA, which can be easily isolated and sequenced for viruses and bacteria. They have small genomes, or a selective choice of characters introduces a lot of bias, garbage in, garbage out. For example, ears, fingernails, social structure would place chimps and humans in the same ramen, but we know that chimpanzees and humans are very different from one another. We need a holistic view of the different characters which describe different kinds. And so therefore another approach is needed to augment morphology, and which is molecular baraminology, which we will talk about tomorrow. So thanks for your attention, and if you have any questions, then please feel free to ask. Thank you very much. Yes.
When God created everything, it was mature, for lack of a better word.
Mature.
The trees were the trees.
Yes, because I mean the birds they flew across the face of the firmament, and Adam was fully formed. He was about maybe 25 years old. Big question: did he have a belly button? I think he had a belly button. So they would so the organisms they would have been fully formed. So if it's the chicken and the egg question, then the chicken came first.
Right. With the ability to reproduce, like the trees.
Yes. Because they multiply according to their kind, so they need their individual sex cells to do that.
I just wonder why did he not? I mean, this is a question that you might have an answer to. Why did he not just sow the seeds and create the baby? You know,
Well,
I don't understand why everything has to be thrown up. And right now, when science looks at trees, it says it's been there for a million years. Because God made it already sure.
yeah, you know that is a good question to think about when you're falling asleep. But I mean, I think that if organisms were fully formed in their fully formed form, they would glorify God by what they do, how they live, how they do their thing. And so I think that that they would have been fully formed. I mean, birds need to learn how to fly, and so but so birds they flew across the firmament. So I think they were adult, fully formed individuals. Yes.
The idea of the holobarams, and they're a kind, like dogs are one holobaram, and cats are another holobaram.
Yes, exactly.
Can there be hybridization between the holobaramins?
No. So cats do not breed with dogs.
Then what about a species like the cheetah, which has characteristics very much like dogs, and it also has characteristics very much like cats. So is it a cat or a dog?
Well, a cheetah. I mean, morphologically, I think that
Yeah.
it looks. I mean, definitely looks more like a cat. I mean, in baraminology, there's use of intuition, because when Adam he named the animals, he could intuitively recognize differences between a horse or I know, like a dog or something. Because I mean every human is a scientist, and so we have this intuitive capability of being able to differentiate between two different kinds.
And there's other individual species like the platypus, which seems to be something all
Yeah.
one thing. But the cheetah, one of the things that they're concerned about is that it has very limited they're kind of going extinct because they don't have much diversity. It's a single.
genetic diversity.
And the thing that makes me think that is, they're the only cat that doesn't have retractable paws. At least that's what the right.
That could possibly be due to a genetic mutation, like a loss in traits. They might have lost the capability to produce a certain muscle which can retract their claws. But you know, cheetah having dog characteristics. I mean, when you look at the organism as a whole, you know, that's why we need a holistic approach to compare, let's say, a cheetah with the dogs. Statistically speaking, if you look at the whole organism, you might find similarities. I mean, cheetahs have fur, dogs have fur. Are they the same kind? They're not the same kind. When you look at all the other characteristics, you'll find that the differences are in the greater majority than there's similarities.
I have one question.
Okay.
Okay, so, when was the first animal DNA discovered or performed? And also, what did that tell us about the animals in terms of, you know, we can't really look at an animal and know about it, but what did the DNA tell us? Was it more profound findings?
So when was the first DNA isolation performed, and what can DNA tell us about an organism? So DNA structure was first discovered in the 1950s, Watson and Crick. When was it first isolated? I mean, the first in animals. I'm not really so sure. Maybe 1980s, 1990s. And of course, now they can isolate mitochondrial DNA very easily because, you know, the mitochondria, it's a cell organelle. So like we have hearts, we have livers, we have brains. Cell has organelles like the mitochondria. The mitochondria has a small piece of DNA. It's 16,000 letters long. It's very abundant, very short. It's easy to isolate and sequence. At this big genetic database online, NCBI is the name. There are like 23,000 mitochondrial genome sequences. I've done a lot of mitochondrial genome analysis. So you can use mitochondrial DNA in Barominology studies when you compare the sequence similarity, or you can do like a whole genome sequencing project, which costs like billions of dollars. The first genome project was Hugo, the Human Genome Project in the 1990s under President Clinton, and the people who did it, they got a Nobel. I think they got a Nobel Prize for it. So if you have the whole entire genome sequence, then you can find the gene sequences, like promoter sequences, like insulator sequences. You know the whole entire genomic information of a given species. They have like the whole genome sequences only for like maybe several hundred species. The best annotated genome sequence is that of human, our own species, because of medical usage. I mean, I use the human genome sequence and its annotation all the time. So there are medical purposes. You can study mutations. You can like find drugs. For example, at a pharmaceutical company, we're studying cancer data. We're studying mutations. And so we wanted to design drugs for individual types of cancers. But I mean, the more genetic information you have of a given species and the better it's annotated, the more you can do. And of course, annotation means that, let's say you get like a raw sequence. You have a string of A, C, T, and G. It's just a raw sequence. You don't really know what it does. You have to annotate it. You have to, by a protein homology, you have to find certain parts of the DNA sequence which correspond to genes, which code enzyme or whatever. So like sequencing the whole genome sequence is one project, but then annotating the whole thing is another thing. It's another lengthy process. And of course, you need algorithms to discover gene sequences. Or what you can do is you can do RNA-seq experiments. You can, for example, if you collect, let's say, cancer sample and a normal sample, you collect the messenger RNA, which is a copy of a gene sequence. You can fragment the mRNA sequence and then you can map it back to the human genome sequence, and comparing the control and the cancer sequence, you can see that the cancer sequence, the cancer sample in XY gene has more sequences mapping to that gene. You can tell whether whatever gene is expressed more highly in cancer as compared to control. So
Endless.
yeah, it's a topic of it's a whole college course actually. Okay, yes.
Did you say you found that there's 4 different bat kinds?
How many bat kinds? According to my analysis of mitochondrial sequences, mitochondrial genomes in bats, I found 4 megabats, Vespertilionidae, Noctilionidae, and the 4th one I forget. I'd have to look at my paper, but I think there are 4. I've also looked at I think the whole genome sequence, comparing like DNA motive signatures in the genome sequence based on these 2 studies, and I'll talk about this tomorrow actually. I think that there are 4 different bat kinds.
Say that COVID actually came from.
So did COVID come from bats? That's a very good topic. Well, I personally think that it's artificial. It's artificial. The communists did it. Next person.
So I hope it's not entirely off topic, but you talked about Neanderthals. And I thought that was entirely a product of evolution. There's evidence that Neanderthals existed.
So did Neanderthals really exist? Yes, they did. Their bones, their skulls, they have a bigger cranial capacity than modern humans. So they're smarter than us, if evolution is true. So I mean, they have thicker bones, like a more elongated skull. They may have suffered from scurvy or gout, which affected their bones. But the thing is that they're an isolated people group. So if they're isolated, there's interbreeding, they're losing genetic information. So that might be the reason why they have these distinct morphological features. But I mean, we've assembled the Neanderthal genome, and they are us and we are them. So we form a reproductive. So since every single person has Neanderthal DNA in them, it can only be because we're a reproductive community. We're the same kind.
Anywhere in Europe, did they say? They existed in Europe?
In Siberia, in Northern Africa, in Israel, Spain, and in Europe, like Eurasia mainly.
Last question.
Yes.
You said that there could never be 1,000-foot human.
Yeah.
Because it couldn't, the blood flow, but yet there's whales. I'm trying to understand when you're just saying for the humans, but animals, I mean, they're really huge animals.
Okay, so why not have a 1,000-foot tall human if they're whales? That's a good point. But okay, we might not use, let's say, this as an example. There's simply certain physical traits and characters which make it impossible for humans to exist in such a state. And I was trying to figure out some example, but yeah. But I mean, like, for example, like what can I say? Like yeah, or what can you say? Like humans with 2 heads, for example.
There are humans who do that.
There are actually the twins. Well, Siamese twins. I mean, they could classify as two separate individuals, but what I mean is to have some sort of very extreme physical characteristic which makes the person inviable, that they can't live in with such characteristics. Yeah. Yes.
I remember when I took zoology in college a long time ago, I got the feeling that scientists struggled with how they classify species, and they still do. They seem to, like bacteria, for example, they rename, reclassify species of veterinary bacteria, for example, that affect animals. So I was just wondering, do you actually get interest from secular scientists in what you're doing? That maybe the look into kinds maybe might be useful for them as they attempt to classify animals into genus and species, or you get a lot of negativity?
Well, I mean, the fact of the matter is that the reason I have to do stealth publications, the reason it's stealth, is because if I use this terminology, Barominology terminology, in a paper, they'll reject it off the bat. And being secular evolutionists, I think they would just make a sport of me rather than taking me seriously. I mean, they don't want to admit the implications of the analysis that I'm actually doing. But I mean, when I read the scientific literature, I mean, I come across monophyletic groups a lot of times. And so this is a code word for Baramin. They use the same concept, but with a different term. Monophyletic group, you know, it means that basically, let's say dogs, the Organo is the wolf. And so they merely say that, oh, it's a monophyletic group. It's a group of species very highly similar to one another. And, you know, quite dissimilar from all other groups, but they're just presupposing, they're connecting the wolves, you know, the dogs with non-dogs. They're saying there has got to be a connection. They cannot be supernaturally, individually, distinctly created. But still they recognize that there's a group there. There's a monophyletic group. I just simply take the evidence as it is. There is no connection between non-dogs. I call it a Brahmin, they call it monophyletic group. Let them be happy. I'll use that term also. It's scientifically correct. But I mean, they're just, it's an evolutionary faith that there's got to be a connecting link between dogs and non-dogs. They've never seen it. There's no evidence for it. It's a religion. It's a religion.
Yeah.
So yes.
Last question, okay, the last one.
Yeah.
All right, I noticed in one of your early slides, it was interesting, you showed that genetic bottleneck at the flood. And the one thing I noticed is you seem to show a more rapid branching with those post-flood species, even with the created ones.
Yeah.
And that was interesting, and also that same applied whether it was a species that might have survived outside the ark versus those within. So that seems to have a lot of implications, including even to what Dr. Jameson will be talking about. So what's your reaction to that? What are your thoughts?
So why do kinds branch so rapidly after the flood? That's a question.
Right, it wasn't like a constant branching rate, but
Yeah.
rather a very rapid branching, and then the tentacles just kind of went up at a slower
Yeah.
branching.
Okay, well, see, the thing is, after the flood, the whole earth was destroyed. So you've got all these environments that animals can invade. They can diversify very rapidly after the flood. There are all these new vistas to explore, all these new environments to assimilate to. So that's why when they discover all these new ecological niches after the flood, there's a rapid burst of diversification, which is happening after the flood. It makes very much sense according to the creation model, actually. But Dr. Jameson can tell us more about this. He's actually written about this whole entire book. So, okay. And so, and of course, I mean, I said that there were some other hands, but afterwards you can come and ask me your questions individually if you have any more questions. So thank you very much.